Dear students, I am Dr. Jamil Abbas from the Physiology Department of Faisalabad Medical University. Here we are going to start a new chapter of physiology, that is the renal physiology, or you can say the physiological aspects of the kidney. This is a very clinical type of the subject, and you have to understand. I think this is the most difficult subject out of your physiology course of the second year. Because this kidney physiology, or you can say the proper word for this is the renal physiology, is very much applied one in your clinical life, and you have to understand all of these things which are concerned in this chapter. We will move step by step. Today's lecture is just an introduction, how we are going to read it, how we are going to study it, and this is your first diagram regarding your kidney. This is your follow my cursor. This on the corner you will find that we have got both the two kidneys which are present on our lumbar regions, and this picture is from the front, and this is your kidney is one kidney, another kidney, and you have got the adrenal glands at the top, or you can say on the top of your kidneys you have got the adrenal glands which we have already studied in the phase of the endocrinology. And here is the cut section of your kidney. You can see you can divide the kidney in two major portions. This part where my cursor is moving now, this hole is known as the pelvis of the kidney. This is the pelvis. Actually, this is the main gate of the kidney, from where any renal vein, your renal artery in the red color, renal vein the bluish color, and the renal nerves in the yellow color, and the ureter. This hole is the ureter. If you can follow this thing, this hole is the ureter. All these things are moving into the kidney or leaving from the kidney. All these are coming through this pelvis. And if you divide this kidney, this outer portion, if you follow my cursor, this outer portion of this whole of this one, this you can see all these things. This is the cortex. The outer portion is known as the cortex, which is a thin as compared to the inner portion. If you see these things, this is your inner portion, and this is known as the medulla. This one is this hole is the medulla. This hole is the medulla, and the outer one is the cortex. This hole is the cortex. And if you move inside the medulla, you will see this is a triangular structure. This is a triangular structure. Again, this is a triangular structure. This is known as the pyramids. These are known because the shape resembles with the pyramids. So these are known as the pyramids. Then if you move onward towards the pelvis, this is your minor calyces. Here you can see the word written over here, the minor calyces. Then all these minor calyces, this is a minor calyces, this is a minor calyces, this is a minor calyces. These are opening into the major calyces. These are opening into, this is your major calyces. So this is your minor, follow my cursor, this is your minor cursor, minor calyx, and this is your major calyx, and these are opening into the pelvis. This all is opening into the pelvis. So this is a brief, you can say, the physiological anatomy of the kidney. We have got the pelvis. In the pelvis you will see the renal vein is leaving from the kidney, then renal artery is entering into the kidney, and then we see the renal nerves which are penetrating the kidney. Then we have got very important structure that is known as the ureter. This ureter is actually the urine formed by the kidney. All this is drained by this ureter into your urinary bladder. We'll see this all in the next you can say slide. But the very important thing, this portion where your pyramid is going to open into the minor calyces, this portion is known as the papilla. This one is known as the papilla. You see here, you can say this is the papilla, this one is the papilla, here this one is the papilla. So we have got the pyramids, we have got the papilla, then we have the minor calyces, then the major calyces. All these are draining into the renal pelvis. And this portion which I am repeating again and again, pelvis is also known as the renal hilum. This portion is also known as the renal hilum. And is the blood supply of the kidney. We'll see in the next slide how the blood is supplied to the kidney. Here the anatomical position of the kidney. This is a frontal view. You are looking at your body from the front. You have got the two kidneys. The adrenals are not shown over here. These are the aorta on one side and inferior vena cava on the other side. And this is the renal blood vessels moving in and out. And here yellowish color is the name, that is ureter. That ureter is draining into the urinary bladder, which is a temporary storage house for the formed urine by the kidneys. The kidneys which are going to form the urine. then ultimately is drained in through this ureter into this urinary bladder, drop by drop, and stored over here, and it can be evacuated. You can pass this urine out of your body when it is required. So this is your urinary bladder, the storage house for the formed urine by your kidneys. This is a theoretical diagram of your nephron. Basically, nephron is a functional unit of the kidney, and this diagram which are displayed on the screen is not the actual one. Actually, nephron is not like this. Nephron is quite different like this, but this theoretical diagram is just to make you understand how we are going to study step by step your kidney. We have to study one nephron, and then this one nephron will collectively we will study that how they are functioning. All this nephron, we will see the physiological anatomy of the nephron. Here you can see this is your afferent arteriole which is draining the blood into this one, the bunch of capillaries, and this is known as the glomerulus. This one is the glomerulus, and this is draining the blood into this one, afferent arteriole, which is draining the blood from the heart through renal blood flow through the renal artery, and ultimately is divided into this capillary network which is known as the glomeruli. Individually known as the glomerulus. The plural of this glomerulus is glomeruli. And this glomerulus is drained out by the efferent arteriole. This is the efferent arteriole which is draining the blood out, and ultimately it is converting into the peritubular capillary network and ultimately the renal vein. We will see all these things step by step, and you will see that surrounding this surrounding this glomerulus, we have got a cup-shaped structure which is surrounding your glomerulus, just making a fist over it. So this is covered by all around from this glomerulus is covered from all sides by the Bowman capsule. This Bowman capsule has got two layers. You will see this if you follow the cursor. We have got the visceral layer. This visceral layer is getting attached with your glomerulus, and we have got the parietal layer. This is your parietal layer which is out of it, and inside you have got the visceral layer. And this arrow is showing that something is draining out, something is filtered out from this glomerulus into this area, and this pinkish one, this initial unit, this pinkish one is known the proximal convoluted tubule. Proximal means near, proximal convoluted means having convolutions. So this one is known the proximal convoluted tubule. And then we have got this your greenish one. This is a loop of Henle, and actually you have to follow this dotted line also. This dotted line demarcates between the cortex above. Here you see the renal cortex above, and this you have got the renal medulla. This is hypotonic area. We will see later on why this is hypotonic. There is more solutes and more salts are over here, and this is having less salts over here. So it's hypotonic and this is hypotonic. And again back to this area. This is your loop of Henle. This is a loop of Henle, and Henle is the name of the science. Is loop of Henle, the loop-like structure. Then we have got distal convoluted tubule. All this is your distal convoluted tubule. Actually, the loop of Henle is divided into two portions. This is a thin part. This is a thin part of the loop of Henle, and here we have got the thick part of the loop of Henle. Here we have got the thick part of the loop of Henle. Then we have got again distal convoluted tubule. This is the light pink. This is a distal convoluted tubule, and this distal convoluted tubule is going to join with the collecting duct, and then collecting duct, ultimately multiple collecting ducts are joined together and they drain the urine formed by this whole nephron into the renal pelvis, and they will excrete it out from the body. So when we study the kidney or the renal physiology, we will move into different segments. First of all, we will study only and only this portion. My cursor is circling out this portion. This is your first chapter, which is concerned with the filtration. And filtration, the kidney, we call it the GFR, glomerular filtration rate. Again and again I will repeat this terminology, this abbreviation GFR. That means glomerular filtration rate. We will study first chapter regarding this area and also the blood supply to this area and this area. Then this chapter is concerned with the filtration. That means most of the things, or you can say some things, they are going to be, I'm not going to name those things, but some things they are going to be filtered out through these glomerular capillaries into the Bowman capsule, and that's all. This thing is we will study in the first chapter. Then in the second chapter, we will see that this part. This is the first part. This is a proximal convoluted tubule. This is concerned with the process of the reabsorption. I will tell you soon that what is meant by the reabsorption, what is meant by the filtration, all the things, the terminologies we will see soon in the next slides. So this is your second chapter. And this whole portion, the proximal convoluted tubule, is concerned with the reabsorption. That means you will see reabsorption means the things we have already filtered over here, they will go back to the blood. The arrow is showing that they are going back to the blood. So this will show the reabsorption. And this will say this whole portion is concerned with the reabsorption. And then loop of Henle. This is your third chapter. This loop of Henle is basically concerned with the formation of diluted urine or a concentrated urine. That means if according to your environment, or you can say according to the season, if you have got a hot environment, that is summer weather, you need to reserve the water, you need to conserve the water actually. So at that time you need to save the water, so you have to pass all the salts, or you can say waste products like urea, with less water. So this is a concentrated urine. This is a function of this loop of Henle. And then if you want to pass a diluted urine, that is same amount of salt but with more water, that is known as the diluted urine. Then this portion, particularly this portion, is very much important. This portion, the arrow, you see 3 arrows are there which is coming out, is very important area from where we actually going to form a dilute urine or a concentrated urine. And this is a hormone which we will study soon, that is known as the ADH. We have seen this hormone in the endocrinology, but we will again study this hormone in the chapter of the kidney. So then we will move to this one. This portion is known as the distal convoluted tubule. Basically, this distal convoluted tubule has got multiple actions, different types of hormones. Although the hormones are also acting on this area, but most of the hormones, they are acting on this area. Hormones, they are also acting on this area. But from here, if you can follow my cursor, from here onward, actually from behind up to all these things, they are approximately, they are in the proximal convoluted tubule. Most of the things, they are mandatory, we have to do all these things. But from here onward, in the loop of Henle, in the distal convoluted tubule, things are according to the things are you can say according to your demands, according to your requirement of the functions. So the function of this area, loop of Henle, function of the distal convoluted tubule, is according to your demands of the environment, of whether you want to pass a dilute urine, whether you want to pass a concentrated urine, multiple other functions we will see here. We will see ultimately what's happening in your acid-base balance. All these things we will see in later on multiple chapters. And this is just collecting duct. It's going to collect that urine formed by this area. So this is a rough introduction of the nephron. Again, keep in mind that this is not the actual picture of the nephron. I will show you actual picture soon, but this is a theoretical one just to make you understand what's happening here. That is, first of all, we study this chapter, GFR, glomerular filtration rate. Then this chapter is concerned with the reabsorption, and then we have got the loop of Henle, which is concerned with the dilute or the concentrated urine. Then we have got the distal convoluted tubule. Here the multiple processes are going to occur. One of very important thing is the secretion. I will make you understand. Here you can see the arrow is from blood vessel towards the nephron. Here you will see the reabsorption arrow is going towards the blood vessel. So, actually we are using 4 terms for the reabsorption in the renal physiology. One of them is filtration. That is, things are moving from the glomerulus into the Bowman capsule, and this is known as the ultrafiltration. because this process of filtration is the pressure difference. Pressure difference is there, and that pressure difference we will see later. There is approximately of 10 millimeter of mercury. And here you will see the arrow is going back to the blood. That means once the things are absorbed, once the things are filtered over here and they are flowing into the proximal convoluted tubule, they can move back to the blood. They can move back to the blood, and this is known as the reabsorption. That is back to the blood. But some of the substances, they are not filtered over here. They can move as it is over here, and they can again come back to your distal convoluted tubule, and this is known as the secretion. Secretion means back to the nephron. And then we use a term that is known as the excretion. That is, excretion means out of your body. So these are the different terminologies we will see later on. So this is a rough introduction that how we are going to study, how we have the plan to study up to finish the renal physiology, GFR, reabsorption, concentrated dilute urine, tubular secretion, and then the collecting duct. And in the first chapter, one thing very important is included in the new, you can say, new version of the guidance, that is the urinary bladder. The urinary bladder is, sorry to say, wait a minute. This urinary bladder is also included in your first chapter. So we will study in the first chapter of the GFR, we will also study the different functions of urinary bladder. So these are the multiple things which we have to study, which we're going to see how we are going to plan, how we have the plan to study the kidneys. So again, this is a diagram of the nephron. Again, you can say that you can see that this is your renal cortex. Cortex is a thin portion as compared to the medulla, which is quite thick as compared to the cortex. Here you see the nephron. This is your afferent arteriole, this Bowman capsule, and this is your proximal tubule. This is your proximal tubule, which is dipping down into the medulla. This is your PCT. Again, in the next, I am using the word PCT. This is your proximal convoluted tubule. That is the PCT. Then we have got the loop of Henle dipping down and deep into the medulla, and then we have got the thick. This is the thin portion, and this is your thick portion. And then we have got the distal convoluted tubule. This is your distal convoluted tubule, and this is the collecting duct which is going to collect the urine not only from this nephron, also from this nephron. And whole of this picture you can well understand that this loop of Henle, this loop of Henle is quite different from this loop of Henle. Actually, the nephron, there are two types of nephron. Some of them are there known the juxta medullary nephrons. Here the juxta medullary nephron. That means the nephrons which are found very near to the medulla. They are actually here, it's not shown, but this thing is found over here. So they are very near to the medulla, and their loop of Henle is quite long. So these are just 15% of nephrons. They are known as the juxta medullary nephron. Then we have got a cortical nephron. You will see the cortical nephron. That loop of Henle is within the cortex. It is not going deep into the medulla. As compared to if you see this area, your juxtamedullary nephron, we have got the nephron quite deep into the medulla. But in the cortical nephrons, your loop of Henle is limiting up to just within the cortex. So these are different parts of the your nephron and their relation with the cortex and the medulla. Then we will move to the very important thing, that the blood supply of the kidney is very important. Here you can see the blood supply is a rough diagram, theoretical one, that you have got this is a renal vein, this is a renal artery. Again, this yellow dark brown color is the ureter. So this is your pelvis, whole this is your pelvis, and this is your blood vessels. The renal arteries dividing and redividing into different branches, ultimately. But the point to be noted over here that these branches which are moving through the medulla, reaching to the cortex, but they are not supplying to the cortex. Very important. Again, I am repeating that renal artery is dividing. I am not going to name the branches. The names are written over interlobar, this and that, arcuate arteries. All these are things are there. But the point to be noted is that renal artery is dividing again and again and crossing the medulla, but is not going to supply any branch or any blood vessel to this cortical area, to this medullary area. This medullary area is not supplying the blood through these vessels. They will reach the border between the cortex and the medulla. You can say corticomedullary border or corticomedullary junction. From here, They will divide in different branches, and we will see later on how they are going to be divided. Here you can see the blood supply of the kidney. This is a rough idea that approximately kidney is receiving approximately above 1 liter per minute, 1100 ml per minute. This is approximately 21 or 22% of the cardiac output which is received by the both kidneys. And kidneys, they are unique in their function that they have got two capillary sets. Here we have got the two capillary set. We also have got the in your hypophysis hypothalamic axis, you have got again the two capillary sets. Here we have got the two capillary sets. We have got Bowman glomeruli. They are again going to divide to form another capillary set which is known as the peritubular capillary network. I will make you understand these things in next diagram. So here you can well understand that a huge blood supply is received by the kidney. That is 1100 ml per minute, 22% of the cardiac output of the both kidneys. Two capillary sets, glomerulus, this high pressure capillary network, and the peritubular capillary network, which is a low pressure. Here the pressure is a high pressure. This is approximately 60 millimeter of mercury. And peritubular, they have got just low pressure, 13 millimeter of mercury, are arranged in series. That one after another they are arranged. Here you can see this is a picture of the nephron. On this side you will see the picture of the nephron. Here you have got the this is your glomerulus and the proximal convoluted tubule. Then this is your loop of Henle. You are moving back, and this is a collecting duct. Then you have got the ultimately these are other nephrons. They are different types of nephrons. So this is a juxtamedullary nephron. This is a juxtamedullary nephron over here. So the name of the artery. If you want to see the name of the artery, you can well see this area. This is a renal artery. This is your renal artery. This is my cursor. This is a renal artery. Actually, if we start from the aorta, we have got that from aorta you have the branch renal artery. Renal artery is going to divide into segmental arteries. This is your segmental artery. You follow this area, this one. This is your segmental arteries. This segmental artery, this is your segmental arteries. From the segmental artery, we have got the interlobar artery. If you see the interlobar artery, here you have the interlobar arteries. These are the interlobar arteries. And ultimately, we have got this the curved branches. This is the curved branches. So again, this is a curved branch. This is known as the arcuate artery. They are making an arcuate in shape. So they are known as the arcuate arteries. These arcuate arteries are dividing into spring-like branches, which are known as the cortical radiate arteries. Here you got the cortical. This is the cortical radiate arteries, and then ultimately they divide to form the afferent arteriole, and they enter into the glomerulus. We drain the blood into the glomerulus. From the glomerulus, they will drain into the efferent arteriole, which is taking the blood. And efferent arteriole is going to convert into the peritubular capillary network. And this peritubular capillary network, in those you can say nephrons which are known as the juxtamedullary nephrons, that is very close to the nephron, where we have got another name of the artery, or you can say capillary, which is known as the vasa recta. We will see in detail in the third chapter what is the role of this vasa recta, but keep this word in your mind, the vasa recta. So efferent arteriole is going to join your pericapillary tubular network, then the cortical radiate vein, arcuate vein, interlobar vein, renal vein, ultimately the inferior vena cava. So you can match these words with your diagram in this way. In the previous diagram, you also see all these things are present over here. Again, this diagram is for you people. You can mark yourselves how the blood vessels are supplying to the kidney, renal arteries. Ultimately, this is the branches. Ultimately, these are the different branches of this cortical radiate branches are there. There is the interlobar arteries, the arcuate. These are the curved arteries, the interlobar arteries. Actually, we will see this slide later on. This is a theoretical diagram just to make you understand our planning, how to study this chapter of the physiology. Here is again a theoretical diagram just to make you understand the different terminologies. I have made you understand through a previous diagram, but this is again very elaborative for you. That here you have got the afferent arteriole, that is blood is drained into the glomerulus. This is your glomerulus, and this cup-shaped structure, and this is your the Bowman capsule. This is your visceral layer of the Bowman capsule, and this is your parietal layer of the Bowman capsule. Here the afferent arteriole, which is draining the blood into the glomerulus, and from here some things are going to be filtered out. Point number one, this filtered out. This is known the filtration process, and this filtration is going, all the filtration, they are going to occur in your cortex, because all the glomeruli, all the Bowman capsule, they are present in the cortex. Then remaining things which are not filtered out here, actually the albumin, basically albumin is not going to be filtered out. Your WBCs, platelets, RBCs, they are not going to be filtered out. Mainly the filtration process for the plasma only. So rest of the blood which is not filtered out will move back into this efferent arteriole. And this efferent arteriole is going to join a network which is known as the peritubular capillary network. Actually, the peritubular capillary network is nothing but the extension of the efferent arteriole. Although it's not this one, this is a theoretical one, it is surrounding this one like this, like rope. It is surrounding in this way, in snake way, it's zigzag way. It is going to surround your nephron in this way. So this is the peritubular capillary network. And here you can see the point number two, that some substances, once they are filtered out, they will move back to the peritubular capillary network. This Bowman, this is glomerular capillary. This is a high pressure capillary network. We have got 60 millimeter of mercury pressure network over here. Here this is low pressure area. We will see later on why it's low pressure, why it's high pressure. This is a low pressure area, and things are, once they are absorbed, once they are filtered over here, point number one, most of the things, they are going to be reabsorbed back to the peritubular capillary network. But at the same time, some substances, I have to mention the name of those things. As I mentioned, that the albumin, WBCs, platelets, RBCs, they are not allowed to be filtered out over here. So proteins, mainly the albumin, is not going to be filtered out over here. But at the end of this chapter, we will see that some of the protein, they do filter out. But at this stage, you will see that no albumin is going to be filtered out over here. But some of the substances, once they are filtered out, they will move back and they will be reabsorbed into the peritubular capillary network. Why it is going to be reabsorbed? One of the very important substance is the sodium. Sodium, 65% of the sodium is going to reabsorb in the PCT. So most of the filtrate which is reabsorbed, which is filtered out over here, actually one very important figure is the blood supplied to this area. Out of what, suppose 100 ml blood is supplied to this area, out of which just 20 ml is going to be filtered out. Remaining 80% is moved onward as it is. And the things which are filtered out over here, they will move back to the peritubular capillary network. A very important substance concerned with this one is the sodium one. But some of the substances, they will come back into the nephron. They will come back into the nephron. This is the process of the secretion, and very important thing which is secreted out. Different things are here, but the most important thing is your potassium. Potassium is being secreted from this area. Although potassium is not secreted in the PCT. This is here you are showing the PCT, but this is theoretical. Potassium is secreted out in the distal convoluted tubule basically. And remaining thing, there's the filtration, reabsorption, secretion, and ultimately we have got the excretion. Excretion means the urine you are going to be excrete out, or you are going to urine is going to be flushed out from your bladder. So excretion or urine is equal to the filtration minus reabsorption plus secretion. This is a theoretical, you can say, although actually what's happening over here. So these are the different terminologies which we are going into the coming chapters. So this is all about the renal, you can say, anatomical physiology of the nephron and our planning how to study. We will see you in the next chapter, same with the GFR. Thank you very much.